This is Unit 3, Part 2, and it's on neurophysiology. Neurons are highly excitable or irritable types of cells. They're quite sensitive to stimulation. When stimulated appropriately, they can generate impulses or action potentials, and these can carry along the length of these projections of the cell body called axons, which can be incredibly long compared to the size of the main part of the cell. These action potentials or impulses that nerve cells or neurons can generate are always, when triggered, always the same in size and nature, regardless of the stimulus. They are a underlying characteristic feature, in fact, of the nervous system as a whole. This is how the nervous system communicates with itself, with neurons communicate with each other, and this is how information comes into the nervous system, how you get processing or information being evaluated in the nervous system, and then this is also how signals are sent out to effector tissues like muscle or glands. Well, let's just talk about a few fundamental physics basics. The voltage is a measure of the potential energy that can be generated by separation of charge. So there is a voltage that is generated when the inside of the cell is negative and the outside is positive. The potential difference is the voltage measured between any 2 points. Current is the flow of the charge between the 2 points, and resistance is the hindrance to that flow. Anything that slows down the flow of that charge is resistance. An insulator is any substance that has a high electrical resistance, and a conductor has low electrical resistance. So, having said that, I can tell you that current, measured in amps, is equal to voltage divided by resistance. The higher the resistance, the lower the current will be. The lower the resistance, the higher the current will be, and voltage has a direct as opposed to inverse relationship with current. Higher voltage, higher current, etc. In terms of current, or so-called electric current in the body, it does not reflect electron flow, all right? It's not like the plug in the wall that if you stick your tongue in it, you get zapped by trillions of billions of electrons. Electrical current, when we talk about a current or flow of charge in the body, we're talking about the flow of ions, charged molecules, not electrons, okay? The electrical potential on either side of a cell membrane, and actually all cells have this, not just neurons, it's called a membrane potential. And it is present if there are different numbers of ions on one side of the membrane than on the other, and if the charges are different. Separation of charge. The membrane itself is what is providing resistance to ion flow. And cell membranes are quite interesting in which ions they will allow to go through and when. It's not as if ions can flow through easily. Many require particular channels, and we'll talk about that soon enough. in fact here. So there are these channels in the plasma membrane. They actually are protein structures that are embedded in the phospholipid membrane of the cell membrane, and they are ion specific. You know, it's not the same. It's not like one channel is good for any ion or electrolyte. In other words, they are quite specific, and they are not necessarily open. There are some that are open, and they're called passive or leakage channels. They're always open, and there's a little bit of leaking of the particular ion or electrolyte that they allow through. But then there are other channels that are gated. They are literally closed, and then under particular circumstances will open. And it's not a permanent open, it can close again. There are three major types of these gated channels: chemically gated, voltage gated, and mechanically gated. The chemically gated ones will open if a specific neurotransmitter molecule that they have a receptor to on the gate, on the channel itself, on the gated channel, there's a receptor specific for a particular neurotransmitter. If it binds, then the chemically gated channel opens. For voltage gated channels, they will open and close in response to changes in membrane potential. And mechanically gated ones will open and close in response to physical deformation of the receptor. So these are the kind of things that you get with on the membrane of cells that can respond to pressure, touch, where you get a physical deformation and then the gate opens or closes. And we're going to go through these because there's quite a bit to each of these different types. In terms of the operation of the chemically gated channels, an example of that would be sodium, potassium gated channels. When they're closed, they are closed when the neurotransmitter that they respond to is not bound to the extracellular receptor. In that case, sodium cannot enter the cell and potassium cannot exit the cell. I should point out to you that for a sodium-potassium gated channel, a channel that when open allows sodium and potassium to pass through the membrane, the key to which way the ion or electrolyte moves is determined. The key fact that determines which way the electrolyte moves is the concentration gradient. They will move always from areas of high concentration to areas of lower concentration. And I also remind you that, and this is very important for you to remember, that there are in all cells, in fact, not just neurons, but there are sodium-potassium pumps operating in the membrane. And these pumps, sodium-potassium pumps, these pumps, which are not the same at all as channels, they're quite different. They use energy to pump these electrolytes against the concentration gradient. So in fact, if this is a cell, sodium is getting pumped out by the sodium-potassium pumps, and therefore the concentration of sodium becomes higher on the outside, so it's pumping against this gradient, okay? And the potassium is being pumped into the cell. So you have a higher concentration of potassium on the inside. If this kind of channel should happen to open, then sodium would rush in. So if it does open, because the appropriate neurotransmitter attaches to the specific receptor on it. If it does open, then sodium rushes in and potassium leaves because of the higher concentration of sodium outside and the higher concentration of potassium on the inside. So that's an example of a chemically gated channel. And here you see this kind of sodium-potassium channel, and you can see that this is in pink or purple, a protein embedded in the transmembrane protein embedded in the phospholipid bilayer, which has a particular specific receptor as part of its surface right there. And when the correct neurotransmitter binds to the specific receptor, right? Receptors are not, they don't randomly bind their ligand. They bind very specific. The specificity is extremely restricted to, meaning, in other words, they're specific for a particular kind of molecule to bind. If they do bind the receptor, then the channel, this chemically gated channel, opens, and potassium rushes out because there's a lot more potassium inside than outside, so it's rushing outwards, and sodium moves in, as I mentioned earlier. So this is one example of a kind of chemically gated channel. And remember that in the background there's always this sodium-potassium pump acting to pump sodium out and potassium in. Okay, let's go to a voltage-gated channel and look at voltage-gated channels. In this case, an example would be a sodium channel we're looking at. It is closed when the intracellular environment is negative. In fact, the normal intracellular environment is fairly negative and stably negative, and in that case sodium cannot enter the cell. Now sodium is a positive ion or electrolyte, and it is attracted to a negative charge, but there's no entry point for it. You have to have some sort of sodium channel open, and in this case, if the inside is negative and it's below a certain threshold level, then sodium channels aren't open and the sodium won't enter. However, if that internal intracellular environment becomes more positive and starts creeping up and reaches what's called a threshold level, okay, and on this graph that I'm drawing, let's make this axis charge, where the upper part is more positive and the lower part is more negatively charged, and this is time. If we have the internal cell charge as being stably negative, let's say that let's put 0 up here. Okay, that's a 0 charge, and anything above it is a positive and anything below it is negative. So if the internal charge for this cell is stably negative, then nothing's happening. But if it starts to come up and hits that threshold level, then what you get is that you get an opening of the sodium channels, in this case of sodium channels, and it says is positive, but it means it should say, although it's a little difficult to sometimes for students to get it right away, you'll see soon enough, but let's call this less negative, moving towards positive rather than positive. Okay, so it approaches, once it hits, remember here is the 0, so it's still negative, but it's come close enough to so that it hits that threshold level. And in that case, the sodium channel is open, and remember, because of the sodium-potassium pump, sodium is much higher in concentration outside the cell, and what you get is a rushing in to the cell through the channel, through the sodium channel, of sodium ions, and therefore the inside becomes even more positive, even more suddenly, and it shoots up into the positive range, and that is called an action potential. That sudden rush of sodium in and change of the membrane potential from to quite relatively positive level. And here's an example of that, where you have the inside negative and a sodium voltage gated channel is closed, and then as the inside becomes less negative, more positive, that's the truth of it. It doesn't become positive, it becomes more closer to the positive, less negative. Then it opens at the threshold, it hits the threshold, it opens, and sodium, of course, will rush in when a sodium channel is open because the concentration of sodium is so much higher on the outside. So these gated channels, when open, will allow ions to move quickly across the membrane. And they move actually not only on the chemical concentration gradient along that, but also the electrical gradient. Remember that, for example, a sodium ion is positively charged, and if the inside, let's say that that's the phospholipid bilayer, and you have a channel that suddenly opens up so that the sodium can rush inwards. Well, it is going to rush inwards not only because it has a higher concentration on the outside, but also because the inside is negatively charged and it will be attracted to that negative charge. Okay? Like charges repel each other and positive charges attract each other. And that rushing in or movement of charged ions, electrolytes, is the current, the actual electrical current. And as a result, the inside becomes more positive, so there's a voltage change across the membrane. And here again, this idea of the chemical gradient is a reference to the concentration gradient, that they move from high concentration to low concentration. And here the electrical gradient refers to the relative charge, and ions will move toward an area of opposite charge. So the key factor is the electrochemical gradient, made up of the electrical gradient and the chemical gradient, the concentration gradient. So these two act together. They have to be considered together when you think about whether an ion will move or can move. And of course, it also depends, of course, on whether channels are open or not. So we talked about this separation of charge, and that normally cells are negative on the insides because of the sodium-potassium pump, and neurons do have a negative charge on the inside, and if they're at rest, then the rest, it's called that negative charge is the resting membrane potential. And a typical resting membrane potential would be about minus 70 millivolts across the membrane. I point out to you that depending upon the source, you can, it's not exactly necessarily minus 70 millivolts, but there's a range around that, and you'll see maybe different numbers in other sources, but that's the resting membrane potential. It is generated because of the different concentrations of sodium, potassium, and other things on either side of the membrane, charged particles on either side of the membrane. The main reason that the sodium and potassium differences are present and maintained is because of the action of the sodium-potassium pump that's constantly pumping sodium out and potassium in, but also because of a difference in permeability of the neurolema refers to the cell membrane of a neuron. So there's also a different background normal permeability of the membrane to sodium and potassium. The membrane always, there are some leakage channels for potassium. So potassium can leak, but sodium cannot get in or out unless one of the channels the gated channels gets opened. As a result of this, what you have, in fact, is, if we're looking, this is part of a neuron. Okay, what they've done actually is they've looked at neurons, in fact, characteristic sort of generalized, and there are many types and many shapes, but I'm just going to give you a general idea of a neuron. It's going to be a cell with many projections from it that can pull in, get impulses from other cells, but it will also have one long extension from it called, and it could be extremely long, and that extension is axon. The nucleus is here in the main cell body. And what they've done here in this diagram on the left side here is they've cut a cross section through an axon. So they're showing this extension of the neuron, but the main point of the diagram here is to show the separation of charge and concentration of different ions, charged molecules. Because of the sodium-potassium pump, you have a higher sodium concentration on the outside than on the inside. Because of the sodium pump, again, you have a higher concentration of potassium on the inside than on the outside. So potassium is higher on the inside, lower on the outside, and sodium is higher on the outside, lower on the inside. See, that's the exterior, this is the interior. At the same time, chloride is higher outside. Negatively charged proteins cannot leak out. They're too large, much larger than these other ions or electrolytes. Many proteins are in fact negatively charged. They're too big to leak out, so there's always this, negative contribution to the overall internal charge from proteins that are negatively charged. In the end, when everything gets summed up, there's a minus 70 millivolt difference across the membrane, with the inside being negative and the outside being positive. And the difference is a membrane potential, resting membrane potential of minus 70. As I told you, there are leakage channels, and there's actually, they're showing in this diagram on the right, they are showing some diffusion or leakage of sodium, but potassium is regularly leaking out, and the leakage inwards of sodium is very negligible. And so with loss of positive charge on the inside, loss of potassium from the inside, plus the fact that you have the negative, and when you're losing positive charges from the inside of the cell, then that makes the interior of the cell more negative. Plus the fact that there are quite a few proteins that are negatively charged inside the cell makes the interior more negative. And the fact that the sodium-potassium pump is pumping out 3 sodiums for every 2 potassiums it pumps in. And so more positive charge is going out than positive charged potassium coming in, and thus that contributes to the development and maintenance of that minus 70 millivolt resting membrane potential. Well, those membrane potentials, resting membrane potential, can change suddenly and generate what I told you was an action potential. And sending out those action potentials are sort of like signals. They're impulses that are sent out along axons, and they're used to, by the nervous system to receive signals from outside, to send signals between neurons, in other words, integrate or process the received information, and, if need be, to send out signals to effector tissues. I remind you that the way the nervous system is set up is that it's receiving sensory information from different areas inside your body and from the outside of your body, and those signals, which are, action potentials, go into the central nervous system where the information gets integrated by processing between neurons. And then, if necessary, a signal is sent out via motor neurons to excitable tissues, effector tissues like muscle and glands. So you need to get a change in resting membrane potential towards the more positive to hit threshold to generate an action potential, which is the signal. And that can be produced if you get that sudden change in membrane permeability, if channels open, and the right channels, or if you have change in ion concentrations. There are two major kinds of signals that can occur. One is called a graded potential, and the other is an action potential. And we'll talk about those in the next few slides. So, first of all, changes can be described in three ways. I would say that, and they involve three sort of the changes that occur in membrane potential are characterized in three ways. I don't know if it's events, but yeah, it's an event. One is that the resting membrane potential of minus 70 millivolts, and again I remind you that this is, the millivolt charge, and this is time. So it's at minus 70 here, minus 70 millivolts, and it changes the inside, and let's say that this dotted line is the threshold, and if it passes the threshold, then you generate an action potential. Anything that deflects upwards towards a more positive charge, remember up here it's positive. Anything that deflects upwards is a depolarization. So this is indicating a depolarization. And then that will trigger action potential, and the action potential is a further very, very rapid depolarization. And then anything that deflects downwards back towards the resting membrane potential is a repolarization. So we have depolarization, a change to become less negative, more positive, and then repolarization, it's going back to the negative. The charge, the membrane potential is returning back and goes down, and even overshoots sometimes, overshoots the resting membrane potential at minus 70, but then goes back to that resting membrane potential, minus 70. This area here, in this area here, that would be called a hyperpolarization. So what we have is, as we go along, and I'll change the color and name it as we go along, we have a resting membrane potential here, then a depolarization, then a very rapid, once it hits threshold, a rapid depolarization, and then now we're into an action potential here and repolarization. All that changes, all those changes in membrane potential above the dotted line are the action potential, and then it's repolarizing down. In fact, you have hyperpolarization, and then back up, small depolarization back up to the normal resting membrane potential. Okay. Now, quite often you have changes that occur that don't reach the threshold required to trigger action potential. These are just little depolarizations because of stimuli that the neuron gets, and you get a minor depolarization, and then it repolarizes back to the membrane, resting membrane potential -70. You can even get a hyperpolarizing stimulus to the resting neuron, and it becomes more hyperpolarized. It may return back to the normal resting membrane potential, but it can momentarily become hyperpolarized or depolarized. This, of course, this hyperpolarization is in the opposite direction from where the threshold is, which is higher up, more towards the positive, and so this would be inhibitory. This hyperpolarizing stimulus or hyperpolarization would make it less likely for the neuron to be able to reach threshold and generate an action potential, and therefore it's an inhibitory kind of signal. Whereas a depolarizing signal, a little upward movement, that's potentially activating, although you would need it to be much bigger to actually hit the threshold and trigger a response. Well, these little changes, these short-lived local changes in membrane potential in different small areas on either side of the membrane, just on the internal side of the membrane, are called graded potentials. So if you have a neuron, let's say you have a cell here, and it's a neuron, so it has its own axon going out, and it has all kinds of they happen to be called dendrites that could receive signals, and it's getting, for this I'll do a different color. Let's say it gets a signal from another neuron, from the axon of another neuron. It gets a signal here. Okay, so there, I haven't shown the whole cell, but there's a signal coming, arriving, and stimulating. Okay, and it's not enough of a stimulus to get enough of a change, but it causes a depolarization in here, in this area here. Maybe it causes some depolarization, but just a small one, a graded potential. That graded potential will decrease as it moves, as you move away in distance away from the origin of the signal in here. As you go along the membrane on the inside of the cell to receive the signal, you'll see that it's decreasing in intensity as it distancing increases, moving away from it. The magnitude of the graded potential varies directly with the strength of the stimulus in this case. You would have to have enough of a graded potential to trigger. You'd have to have a strong and you'd have to have enough of a stimulus to depolarize the receiving neuron to the level that it hits the threshold and initiates an action potential. If it's not enough, it's just a graded potential and it fades away. And here you see this, depolarizing effect. In this case, the stimulus is not from another neuron, but from a electrode that's has a charge and touches the plasma membrane. Notice all these charges are shown just near the membrane. That's where all the electrolytes or ions that are affected are. It's just along the surface of the membrane inside the cell and directly outside. Okay. So here you have an electrode and it touches. Maybe it activates a few gates and you get some inrushing of sodium and positive charge here, but not that much. And it fades as it moves away from, the depolarizing effect spreads further away, but it's just a small graded potential. It's not a strong stimulus, so it fades as it spreads away from the site of stimulus. So the voltage change with graded potentials is actually related to the intensity, but if it's not enough to hit the threshold, it'll just quickly fade away as it moves away from the site of. Okay, so here's an example of a graded potential. Let's say the the threshold is up here. This would be threshold, but this is a graded potential that doesn't reach that threshold. We're looking at along this axis now, we're looking again at the charge membrane potential, and it's the resting membrane potential minus 70. Now along the horizontal axis, we're not looking at time, but distance from the site where it was stimulated. And it's showing that the size of the depolarizing effect on the resting membrane potential, this going toward more positive, fades away as you move further away from that initial active area of the depolarizing effect. Okay. Now, as I said, if graded potentials are large enough that they add up together to depolarize the cell, the interior of the cell enough so that it hits threshold, then you generate an action potential. So an action potential is a very brief but intense reversal of the membrane potential. The height could be about 100 millivolts. It actually goes up to into the positive charge range, so the interior of the cell momentarily will become positive near the membrane. They are generated in neurons, but they're also generated in muscle cells. Both muscle tissue and nerve tissue made of muscle cells and nerve cells, neurons, are excitable. In other words, they can generate action potentials, and they're the only types of cells or tissue that can generate action potentials. These do not decrease in strength over distance. There is no change. An action potential gets generated and it will be propagated or sent out in one direction along the axon. If we're talking about neurons, and we are now because we're doing neurophysiology here, not muscle yet. So if we're looking at neurons, the action potential will get sent out down the axon from the main part of the cell, the central cell body where the nucleus is. They do not decrease in strength at all over the distance, and they are the impulse that this particular neuron we're talking about in an example sends out in order for it to send a signal to another cell. They're the principal means of neural communication. So that action potential that gets sent out along the axon of a neuron is in fact the nerve impulse that we talk about when we talk about nerves sending signals or impulses or activating signals. Here we see a portion of the membrane of a neuron which is in the resting state. In other words, it's at the resting membrane potential. The sodium and potassium channels are closed. So if we're looking here at a sodium channel, it's closed. The sodium can't go in. Here we're looking at a potassium channel. It's closed. Potassium can't move outwards. All these channels are closed. There are inactivation gates. There are activation gates that are closed, and that's what keeps things shut. And there are also another type of gate called inactivation gates, and they're open, at least for sodium. So if we just talk about sodium here, looking at sodium, the sodium channel, it has an activation gate and an inactivation gate, and the activation gate is closed. Activation gates are closed in the resting state, and activation gates are open in the resting state. And resting is at the resting membrane potential of -70 roughly. Now, with depolarization, sodium permeability increases because channels start to open. Okay? If you have a depolarizing event, it will allow voltage-gated channels to open, and you have these sodium leaking in or moving in through the channel, and the membrane potential can in fact hit threshold. If it does reach threshold, and then you get a reversal, rapid reversal. Sodium gates open. The potassium gates remain shut, but sodium gates open, and because of the electrical chemical gradient, the sodium will start rushing through to the inside of the cell because of the higher concentration of sodium outside, and because it's a positively charged ion and it's moving towards the negatively charged interior. It hits threshold, and threshold can be at -55 to -50. Remember, the resting membrane potential was at -70, so it's become less negative, more positive, in other words, but less negative is an appropriate way of saying it. So if it does reach that threshold, then the depolarization event becomes self-generating, and it doesn't need any more stimulus to depolarize more. It goes boom, really fast, really suddenly. So this is depolarization reaching threshold. And now you have a big surge of entry, and that's the action potential. That's the depolarizing upward movement because of lots of sodium channels open, and then sodium inactivation gates shut. This inner gate, inside, outside. So it's the inner gate, it's the inactivation channel. Suddenly the permeability of the membrane to sodium declines back to the resting levels. And now, as those sodium gates closed, because of the inactivation gate, voltage sensitive potassium gates open. And now potassium rushes out. Well, if you no longer have sodium rushing in, and now you have potassium rushing out, well, then the inside of the cell is going to start becoming more negative, right? You're losing positive charges on the inside, so it's becoming more negative. So it is repolarizing. So now you have a repolarization, and the resting membrane potential gets restored. It even can hyperpolarize. The potassium gates are still open. You get an excess outward flow of potassium, and so you get some hyperpolarization. During all this time, by the way, you can't stimulate the neuron again to depolarize back to threshold and generate another action potential. It's insensitive to any stimulus at this point. Anyhow, now it's hyperpolarizing, so there's a hyperpolarization phase or hyperpolarization period, and then finally it goes back to the normal resting membrane potential because of the action of the sodium-potassium pump. The sodium-potassium pump will, we had the resting membrane potential and depolarization hitting the threshold, and some rapid depolarization. There's the action potential, then repolarization and hyperpolarization, and then because of the action of the sodium-potassium pump, it will return the neuron to the resting membrane potential. So it restores the resting electrical conditions of the neuron. That repolarization, even the hyperpolarization, all that gets corrected. The imbalance that results is because of all the events that I talked about will get restored by the action of the sodium-potassium pump back so that it goes to this resting membrane potential of approximately minus 70 millivolts. And here you see all of those events shown in this diagram, where you have the resting state or resting membrane potential, which depolarizes up to threshold, the dotted line. Remember, this now is membrane potential. Well, it always is. This is time in milliseconds. There's the 0, so minus 70, but then it hits the threshold. Sodium permeability suddenly rises. Because sodium channels are opening, voltage-gated sodium channels are open. There's a big rush inwards of sodium. Then sodium inactivation gates close and sodium permeability drops, but potassium permeability rises, so potassium is rushing out, and that's the repolarization phase. Here is the depolarizing, the rising. Here is depolarizing phase. So now we're in the repolarizing phase, and it goes down. It even shoots over deeper. It's more repolarizing, so it becomes, you get hyperpolarization, and then what returns it here to the normal resting membrane potential of minus 70 is the sodium-potassium pump. That's an action potential. And that's the end of Unit 3, Part 2.